Hello, good afternoon. Looks like it's just the three of us for now. Thank you for being on time. We're going to go
It's
ahead and
all
get
good, sir.
started.
Thank you.
Okay, great. So, let's do a quick recap of what we've learned last class, last week. We were talking about DNA fingerprinting, but then we actually didn't get to finish the whole story. But along the way, there are some key concepts that we need to understand. So first, there is this thing called the short tandem repeats or STRs. So these are nucleotides that are repeated several number of times depending on the person. So it could be like CCT repeated 10 times, or it could be like CG repeated 5 times. And that in total would be CCT, CCT, CCT repeated 10 times. So that's 10 times 3 nucleotides, giving a total of 30 nucleotides in length. And this one is just 2 nucleotides long, repeated 5 times. So that's going to be 5 times 2, which is 10, 10 nucleotides in total length. And what's important to understand is that these STRs, these short tandem repeats, are going to exist at the same location for everybody. And what's difference between the STR in different people is the number of repeats of the STR. So you're gonna have CCT repeated 10 times. I might have CCT repeated only 5 times, right? People who are related tends to have tandem repeats that are repeated the same number of times compared to people who are not related. So what we need to do is to find a way to be able to separate these different DNA lengths by size. So, if we can um, have the longer ones at one place and the shorter ones at a different location, that would help us create a DNA profile for each individuals. And that's where the technique gel electrophoresis comes in. So gel electrophoresis. Right? So this is a technique used to separate DNA based on its size or length. Okay? It means the same thing for us. So let's say you have a DNA sample, which is a mixture of like in there are some long ones, medium ones, short ones, right? It's like that, all different lengths, right? So these are all DNA, and it's all mixed together. Oops, it's all mixed together in this sample. And to separate it, we are going to put it on a gel. Right? So to draw a gel, it's like drawing a castle. You have a little bit of these uh, kind of teeth looking thing at the top. And that's going to be the well that you're going to be loading the sample in. Of course, this is going to be in the chamber that I showed you last time. One end is going to be hooked up to the negative terminal. The other end is going to be hooked up to the positive terminal. And you will be running a current through it. All right, so current pushes DNA down the gel. Now remember, once you put the sample in, the sample is going to stay in its own lane. All right, so think of this as like, Olympic swimming competition. Right? The square, the rectangle here is where you load the sample, and then the sample is just going to go down its respective lane like that. Okay, like that. The sample over here in lane number one is not gonna, go diagonal like that. Okay? So, you would run the gel, you would let the current go for, an hour, hour and a half, depending on the experiment you're doing. And what's going to happen in the end is the longer DNA tends to be closer to the well because they have a hard time weaving through the gel, which is like a network of mishmash fibers, right? And the smaller ones, the shorter DNA, are going to be able to migrate further down the gel. So in the end, each sample is going to create its very distinctive banding patterns. And so that's one sample, and then here you might have another sample. Okay. And then based on these banding patterns, it's almost like the person is going to have like a barcode that's unique to them. And you can compare the barcode of one person, compare the DNA profile of one person with another. Right? So that's basically what electrophoresis is. Now, exactly how you create these bandings, I'm going to go through that process with you. Okay. But that's all the stuff that um, is important in terms of helping you understand the next portion of the note. And these are things that we did on Wednesday last week. All right, so here we go. Let's talk about STR analysis. Okay, so STR analysis is one form of DNA fingerprinting, and it's useful for matching a suspect to a crime scene sample, or we can use it to determine relationship between individuals, like is this your father, or like is this your long lost sibling? Right, all these things can be done using STR analysis. So if you have three different colors, it would be ideal for taking the following notes. Okay, four colors actually, okay, including black. So here we have a person. Okay. Okay. This is gonna be Bob. All right. And for Bob, we are going to be looking at three of his chromosomes. For simplicity's sake, we're just gonna look at three of his chromosomes. Okay, I'll explain how it's done in real life at the end. So on chromosome 7, he has an STR, which I'm going to use red to represent here. And this STR is CCT repeated 20 times. And then on a different chromosome, chromosome 10 or something, there is going to be another STR. This one is TA repeated 5 times. And then over here, maybe we have ACC repeated 10 times. And let's just say this is chromosome uh, 20, okay? Doesn't really matter. So I'll give you a moment to copy all of that. All right, let's take a look at another person here. Okay, this is gonna be Jill. Okay. And Jill, we're gonna look at the same chromosome for her. Okay, same 3 chromosomes. So because Bob has an STR on chromosome 7 at this location, you would expect everyone to have the same STR on the same chromosome at the same place. But Bob is not related to Jill, so their STR is probably going to be different in lengths. Okay? So this one, I'm just going to say it's CCT25. It's repeated 25 times instead of 20 times. And on chromosome 10, all right, she's going to also have the TA. This one happens to be the same. Maybe they are, of the same ethnic background. And so there are some similarities in terms of the STR on some locations. And then over here, she's going to have an ACC, and this one's going to repeat. Hold on, let me think about it so that it's going to work out. Let's do 5 times, okay? 5 times. And this is going to be also chromosome 20. Okay, nothing to understand so far, I suppose. It's just, examples that I randomly make up. So we already know all the STR locations in the human genome, right? Because of the Human Genome Project, right? You can actually go find out where these repeats are for everybody. And we can design primers to clone them. So I can have a primer that binds to, before and after this region. And then I would have a primer for the blue one, and then primer for the green one here. And then we would use PCR to clone these STRs. So you would get a DNA sample from Bob, do like a cheek swab or something, like blood that has that DNA in it. And then you would use the primers here, the red one, the blue ones, and the green ones, to amplify, to make many copies of these STRs. You're not going to copy all of the DNA. You're just going to copy the selected STR. And we're only doing 3 of them. So at the end, you're gonna have a little one of those little tubes that I showed you last time, and it's gonna have, the red, many copies of the red STR, many copies of the blue ones, and then you're gonna have many copies of the green ones, all mixed together. Okay. Of course, it's just gonna be colorless. You can't really see it. You can't really see the actual DNA with your naked eye, but it's gonna be in this tube. Okay. And then we're gonna do the same thing here for Jill's sample. We're going to do a cheek swab for her as well. And then we're going to use PCR to clone these STRs. Same thing. Okay? Same primer, okay? Because you already know where they are. So when you design the primers, they're going to flank the specific STR. And in the end, you will have a separate tube for Jill's DNA. Now let's take a look at here. It's repeated 20 times, and it's 3 nucleotides for the triplet here. And so the total length of this STR is going to be 20 times 3, which is 60. For Jill over here is 3 times 25, which is 75. Over here, 5 times 2 is 10, and here as well. 3 times 10 is 30. 5 times 3 is 15. Okay, so this DNA is 60 nucleotides long. This is 10 nucleotides long, so on and so forth. And you just multiply the number of nucleotides in the bracket by the number of repeats. And so let's draw a gel here. All right, so there's the gel for electrophoresis. Okay, we're going to draw a little bit of a teeth mark up at the top. That's the well to load your sample in. So let's put Bob's sample here. Okay, so this is Bob's lane. Right? His DNA, the one that we copy, amplify, is gonna run down this lane. And then we're gonna put Jill's DNA right next to his so that we can compare it easily. And her DNA is gonna run down this lane. Okay? Remember, no one's gonna be going sideways, not gonna be going upwards. Okay, just gonna go down the lane. And the different fragments are gonna get separated based on its length. So let's take a look at Bob's DNA here. Remember, when we do it in real life, it's not going to be color, right? In fact, you can't see it until you actually stain the DNA. I think maybe I haven't show you that picture yet. Let me give me a sec. Give me a second here. So after you run the gel, like I showed you in the video last time, the only thing you can see is it's the blue line, right? Which is the dye front, and that's not your DNA. That's just a dye that gives you an idea roughly where the DNA are. To actually see the DNA, you need to stain it with something called ethidium bromide, which binds to the DNA and makes it glow when you shine UV light at it. Okay? And then you can take a picture of the gel, which turns out to be black and white like that. So that's the DNA sample right there. Okay, I mean the whole sample is loaded in lane two, but there is only one very strong band that tells you most of your DNA is going to be quite short in this case, right? And then here in lane four and five, you have DNA that are longer than this one. Remember, this is negative, this is positive. So the shorter it is, the further it can travel down the lane. Now, in lane number nine, and then one here as well, you can see there are multiple bands, right? So it's actually common practice, and you can see it over here as well. It's common practice to load a ladder, it's called DNA ladder, at first lane and last lane for comparison. Okay, so a DNA ladder are basically a prefabricated mixture of DNA that you know their length size. Right? So you can buy this from companies, right? And so when you load it, you have DNA fragments of different size um, showing up here. So when you run your own sample and you see a band here and you want to know, hey, how long exactly is this? Then you compare it to the ladder, and then you will be able to figure out the exact measurement. Okay. Now back to our picture here. So we're looking at Bob's sample, and this is 60 base pair long. Is that going to be closer to the top of the gel or further down the gel? What do you guys think? Closer to the top or further down the gel? Now we can see there are three bands here, right? 60, 10, and 30. Since 60 is the longest one, is going to be closer up here. Okay, it's the longest one. 10 in total is going to be furthest down. Right? It's the smallest one, so the current's going to be able to push it through all the way to the end. And over here, it says 30. 30 is going to be somewhere in between here. It's not exactly halfway because these things are not, it's not like linear. It's not like 60 up here, then 30 has to be exactly halfway. It doesn't work like that, right? So there you go. So that's the three bands for Bob. Now let's take a look at Jill. Jill also has the same three STR position, but for her red one, it's repeated 25 times instead of 20 times, so it's a little bit longer. It's 75. Do you expect Jill's red band to be same level as Bob, higher than Bob's, or lower than Bob's? Think about that for a second. Yeah, it is definitely going to be higher than Bob's because 75 is longer than 60. It's going to take a long, like it's longer. It's not going to be able to go through the gel as easily. So that's going to be the 75 band. You're going to have the same length for the blue one, so they will be at the same place. And last but not least, for the green one, it's repeated 15 times. So 15 is pretty close to 10, um, but it's going to be just slightly above, right? So there, with three different STR locations, we are able to have a distinct banding pattern for Bob and Jill. Okay, and that's basically what STR analysis uh, uh, is about. And if we consider multiple STR, not just three, but multiple STR, then what's going to happen is, each person is going to have a very distinct banding pattern. Okay? So it creates almost like a barcode for each person, right? And remember, there are like 8 billion people on this planet, right? We cannot just use three lines to distinguish them. Right? We have to use as many STR as we know about and, are practically able to clone at the same time. And then we create like a DNA profile. All right. So if we go back to here, in do we have a oh there we go. All right. So that's like a crime scene DNA, and then we have three suspects. Each of these bands again represent our STR, and some are like thicker than others. It just means that maybe you have a few bands that are really close to each other. Like maybe this is 30 and 35 and 40 kind of all close together. It's really hard to separate them. But what's really clear is suspect number two was definitely at the crime scene. Okay, because you can see it's a perfect match, right? So this is very accurate. It's if you get a different banding pattern, then this is not their sample, right? Does that make sense? Okay. I mean, like it shows that number two was there, but it doesn't show that one and three were not there, right? It just means that we didn't pick up their DNA there. And so like it's a little bit interesting there in terms of how you interpret the results. Okay, let's take a look at some of these questions. The plasmid is treated with equal R1 and BAMH1 when the digested plasmid, digested means after you cut it with the restriction enzyme, is separated using gel electrophoresis. How many bands will you see? Well, if you use equal R1, it will cut it right here. And then if you use BAMH, it will cut it right here. And so that's one fragment, and that's another fragment. Call this one and two. So you will have two bands. Number one, band number one over here is shorter, so it will travel further down the gel. Number two is longer, which means it's going to be closer to the well. Okay. Here's the question. There are two DNA samples with an STR polymorphism. It just means their STR are different, right? One is repeated 10 times in sample A, the other repeated 15 times in sample B. Which sample, A or B, will be further down the gel when separated using electrophoresis? So the key thing to remember is that things that are bigger, longer, will stay closer to the well. If it's shorter, it's going to be able to go further. So this one asks you which one goes further. It will have to be sample A because A is shorter. If it's shorter, it's able to weave through the the mishmash, the fibers easier, and therefore travel further. Okay. B is not shorter. Is repeated 15 times. 15 times 3 is 45. 10 times 3 is 30. Okay. All right. Over here, we have a ladder and we have some fragments. Okay. If the DNA sample only has two recognition sites for restriction enzyme, what is the original length of the DNA sample? Okay, you gotta understand, like if you have a linear DNA and you chop it two times, you get three pieces. If you have a circular DNA, like a plasmid, and you cut it two times, then you get two pieces. Right? So depending what your original sample is, the number of fragments you get is not always equal to the number of cuts. But in any case, over here, this is the ladder. That's like a mixture of samples that you already know the individual length. And then over here, you have your three samples. So, sorry, your one sample with three fragments. Um, so this one here, how long do you think A is compared to the ladder? Give me a number here for A. Yeah, that's like somewhere between 10 and 8 kilo base, right? So that's going to be 9 kb. Yeah, very good. All the answers coming in. Over here, this one is no brainer. That's 6 kilo base. How about this one? Yep, that's 2.5. So if you add it all up, 9 plus 6 plus 2.5, that's going to be 17.5 kilobyte kilo base in total. Okay, so like 17500 base pair. Okay. Which is like a normal unit that we use to compare. Okay, that's the example I gave you where it's like only 60 base pair or like, 30 base pair. That's like, you know, it's not really realistic, but I try to use small numbers to help you understand. All right. RFLP analysis. That's another kind of way to check to see if the samples are the same. Basically, you treat the DNA sample with some restriction enzymes, and then you analyze the fragments that they produce. Okay, so let's say there is a restriction enzyme that would cut CCGG. Right, so at the crime scene DNA, the sample has two CCGG, and so the restriction enzyme would cut it once, cut it twice, and then it would produce three fragments. Okay, so that one goes right here. Now, different people have different silent mutations, right? We talk about this, way back in lecture 7. So it's possible that this person has a silent mutation that caused the CCGG to mutate to a CAGG. So this point mutation causes the recognition site to disappear. So when you treat this person's DNA with the same restriction enzyme, it will only cut it once. Right? It's not going to cut here. And now you only have two fragments instead of three. So that's how you can tell, oh, these are different DNA from different people. Right? Because if it's the same person, they will produce the same bending pattern. So the point is to analyze two individuals' DNA. You can do STR or you can use this thing called RFLP. Right? It really depends on what's available and what exactly are you trying to achieve with the comparison. Right? Okay. I don't think you guys have this slide. Uh, I just added it recently. I want to show you how is like actually done, okay? So this is a family, okay? Family, two parents, two kids. Two parents, two kids. Yeah. So we want to figure out who are the dad, the mom, dad, and who are the kids, okay? So which of the following statement is consistent with the results? Okay. You already know that these are families, okay? That's the premise. So let's take a look at the first one here. B is the child of A and C. So A and C, mom and dad, doesn't matter who's who, right? So let's see. If B is the child of A and C, then all his STRs is gonna come from either mom or dad, right? So let's see if that's possible. Okay, so the first band here was inherited from C, right? Because A doesn't have it, right? So this one has to come from C. Now what about this one? Well, this one also came from C, right? Because A doesn't have it, so that's okay. And how about this one? Well, C does not have it, and A does not have it. So where did it come from? I can't explain that. That means, well, okay, I mean, it cannot come from D because like we're just looking at, ABC's relationship, right? So B has a DNA fragment, sorry, has a STR that A and C does not have, which means B is not the kid. Okay? B cannot be the child of A and C because he has a gene that A and C do not. Okay, remember, all your genes has to come from the parents, right? You cannot have a gene that neither of your parents doesn't have. Okay, so that means this is wrong. Wrong, okay? Now let's try another one to see. I'm just gonna try C, okay, because I know the answer. All right, so D is the child of B and C. So B and C. Let's see if D can be the child of B and C. Okay, so let's see. This one here, right? Where does this line come from? Is it from B or is it from C? Neither, right? None. Oh, so that's wrong. No, that's not possible. Okay? Because again, you cannot have a gene that your parents don't have. So that's not. So let's see B, if B is correct. Okay? C is the child of A and B. So A and B. Can C be the child of A and B? Well, let's see. This one here came from B. This one here came from A. This one came from B. This one came from A. This is from A. This is from B. This is from A. This is from B. You just match it up, right? This is from A. And this one is A or B. So everything checks out. Everything in C came from either A or B. All right. And so, yes, C would be the child of A and B. Do you guys understand? Yeah, so that's how it's done in real life, okay? Except, they probably have computers to do it for you. Yeah, it's quite simple, right? And this would be useful for, determining if you found your long lost twin. People do that, right? Like, I actually watch a few Netflix documentary on this. People do their DNA testing like 23andMe or something. There are a few companies for it, and then they take your sample and then match it against their database, and then people find out, oh, I have a like a half sister or something that I didn't know about, or I found my long lost twin, right? And that's the kind of DNA analysis that would be revealing because siblings would have similar banding patterns, okay? And then as you see here, parents and their kids, um, they would also have certain similarities, right? Um, and these kind of similarities would not occur for people who are completely not related. Okay, so I hope that was interesting because it's like a actual, you know, application in real life. Oh, okay. Yeah, yeah. Someone just share an interesting story. Yeah, yeah. Actually, just quickly, again on Netflix, I saw this documentary. There were two twins, okay? Two twins. I think this was at Colombia or something. Let me know if you have also seen this documentary. So two twins, identical twins, okay? Red is identical to red, blue is identical to blue. They are all boys, okay? And then what happens is there was a mix-up at the hospital, okay? So like they swapped the babies. Yeah, I think what happens was like one of the twins was at the hospital, and then the other set of twins, one of them was sick, so the aunts took the baby to the hospital where the other twins were. But then somehow the like the identifying bracelet fell off, and then I guess someone put it on wrong, right? And then the aunt didn't know she took back the wrong twin, the wrong baby. Okay, and then I guess the mother didn't know either because babies they all look similar, right? When they're newborn. And then what happens is so these get raised together at one family, and these gets raised together at another family. So for their entire life they thought that the other child is their non-identical twins, right? And then when they're like 30 or something, one of the brothers, I guess, saw someone online that look exactly like him, which turns out to be his identical twin, right? And then when they meet up, they found out that their brother also looked the same. And then finally they realize, oh no, like there has been a mixed up at birth. And it was really, really weird. And for them, it was traumatizing as well. Finding out, the person who you grew up with is actually not your twin, and then you have another twin out there. That's, you know, uh, bizarre. Anyway, I forgot what the name of the documentary was. I'll post it online if you guys are interested to watch it. Anyways, we are almost done. Oh, there are more questions you can try to figure out yourself. It's not going to be on the quiz, okay? But I thought I'd just show you because it's like I said, interesting to know. All right, finally, gene therapy is the last bit of biotech that we're gonna talk about, and it's very promising in terms of being able to fix a lot of the incurable diseases and conditions that exist right now, and we are at the brink of really cracking the code for gene therapy, and if they are successful then many diseases will that are currently not treatable will finally have a cure. So the basic concept of gene therapy is fairly straightforward. It involves introducing genetic material into the person's cell, and depending on what kind of cell you're introducing the genetic material into, it's either a somatic gene therapy or germline gene therapy. Okay. You learned somatic versus germ cells from me previously. Somatic cells are bodily cells. So if you are modifying the genes of a bodily cells, only you will be affected by the modifications. They do not affect the sperm and the eggs, and therefore, the changes is not going to um, affect future generations. However, if the modification is done in the germline, then you are not going to be affected, but all your offsprings will be affected by that change. So germline therapy is considered unethical by the scientific community, and it is not currently being researched. And for somatic gene therapy, it's approved, and in fact, there has been several successful cases of somatic gene therapy where conditions like sickle cell anemia are being reversed and treated. Okay, which previously had no treatment, right? So the goal of gene therapy is essentially to replace a non-functional gene. It can also be used to suppress an overly active gene or to modify the clinical course of a condition. So let me go into a bit more details for each one of these things. First one, very straightforward, replacing a non-functional copy of a gene. So let's say in sickle cell anemia, right? We talk about sickle cell anemia several times in the course. It is a missense mutation that cause you to um, produce a misfolded hemoglobin protein, and that in turn causes the cells, the red cells, to become moon shaped. They cannot transport oxygen for you, and you suffer from shortness of breath, chronic tiredness, so on and so forth, right? So it's quite a debilitating condition, and it's all because of a single missense mutation. And so if you are able to deliver a functional version of that gene, okay, that does not have the mutation, then the cell is going to function normally. Okay? So when using gene therapy to treat these conditions, the condition usually has to be recessive. Okay? Why? Because if the trait is dominant, when you put in the normal copy, it's not going to matter because the dominant mutation will cover up any non-dominant genes that you insert. Okay, but if it's a recessive condition, inserting a functional gene, the functional gene would be the dominant one, and it would cover up any mistakes or any copies that contains an error, and then you would be able to fix the problem. Okay, so first goal is to replace a non-functional gene. This is only successful if the effects of the disease are reversible or have not resulted in lasting damage to the body. If the disease, let's say the person already has the condition for 10 years, and during that 10 years, because of the mutation, it affected things like brain development, right? And then you give them a successful gene therapy at the age of 10, all those damage that were done in the first 10 years, you cannot reverse it. Okay. So, moving forward, you can prevent further damages, but you cannot undo the damages that already happened, right? So the conditions like those might not be too ideal for gene therapy, at least not at this stage, because the benefit might be marginal. Okay. It's not going to be very impactful. But for things like sickle cell anemia, which, like I told you, is a missense mutation, or beta thalassemia, that's a nonsense mutation in the hemoglobin. These things, yes, they have negative impact on a person's life, but the damage is not irreversible. As soon as the person starts making um, good hemoglobin, then you're gonna start turning out good red cells, and then the problem will be gone. Okay, hemophilia, the bleeding disease, where they are missing a blood clotting factor. So the gene that makes the protein that helps them stop bleeding is not there. Well, if you introduce a functional copy, if you give them the missing recipe to make that protein, then moving forward, you're not going to have the bleeding condition anymore, right? So that's the first goal of gene therapy. Second goal is to suppress a gene, suppress an overly active gene, such as an oncogene. If you remember from lecture 7, an oncogene is a mutated green light, so to speak, right? This is a green light that's always on which allows the cell to keep on dividing and dividing, creating the tumor or potentially creating a cancerous a tumor, right? And so if you can insert a blocking gene that would like cover up the green light, right? So to speak, then you stop that uncontrolled cell division, and you can slow down the tumor growth and combine this with other treatments like surgery, chemotherapy, radiation therapy. Then you have a better chance at surviving the disease than you otherwise would. Third goal is to modify the clinical course of a condition. Right? So the aim here is to insert a DNA into cells that contains the mutation and either trigger cell death or it creates special markers on its surface for white cells to recognize them. Okay, that would be ideal for treating certain types of cancer. So here we have a cancer cell, disease cells, and we are going to deliver the suicide gene. As soon as the suicide gene goes into the cancer cell, it will produce a toxic product and then it will kill the cell. Now, the challenge here is like I'm obviously we're like simplifying things a lot, but you can understand the major challenge here is how do you make sure the suicide gene is going to go into the cancer cell and not your healthy cells, right? Like if the suicide gene goes into your healthy cells and start killing the healthy ones, then it's going to make it worse, right? So right now, the technology is not really there yet. Okay. It's kind of like a hit and miss sometimes. And there are several cases where they try to insert one of these suicide genes, and then the cancer starts to improve, but then the liver stop functioning. Okay, because of the we call these like off-target effects, right? So in order for because liver is very important, right? It, like filters your blood and everything, and so sure, like you're a cure of the cancer, your cancer is not progressing, but then your liver is killing you. And the only way to fix the liver problem is to stop the therapy. And as soon as you stop the therapy, the liver is functioning now, but then the cancer starts to go back, right? So like there's a delicate balance in terms of like therapy and the side effects, right? And we're not quite there in terms of controlling the specific outcomes just yet. Another option is to insert a marker gene. The marker gene puts special markers on the surface of the cell, right? And when an immune cell sees these markers, it will attack them and kill them. Okay? So this is something we'll talk in a great deal in the next semester. All right. I will be teaching one of the second semester classes, so maybe I'll see you again. There are two options with gene therapy. It can happen ex vivo versus in vivo. Vivo is the body, so either you do it outside of the body or you do it inside the body. So let's talk about ex vivo first. Ex vivo involves taking the cells out of your body. Right? For example, we can take cells out of the bone marrow. That's where you will find the white cells. And in the lab, in the petri dish, you're gonna modify these cells. Okay? You can modify them, you can train them to recognize cancer cells, for example, and then you will put it back into the person. Right? And then they will seek out the cancers and then kill them, right? So that's a little bit more controlled because you know exactly what you're doing to these cells, and you're only doing to the cells that you've extracted. You kind of leave the rest of the cells in the body alone, and then you put it back in. So you have more control here. In vivo is to use something to deliver the DNA. Let's say I have a functional gene I want to deliver to the bone marrow to make healthy hemoglobin, then you would use a virus or something to deliver it. Because we talk about how viruses work earlier in the course. Virus actually insert their DNA into your cell's DNA, right? So it's kind of perfect to have some of these as vectors. Because if you give the virus a good copy of the DNA and you infect the person with the virus, theoretically, the virus would go insert that into your genome, and then now you would have a functional copy. Is that bone marrow is for leukemia? Yeah, yeah. So the bone marrow, leukemia is basically blood cancer, right? It has to do with cells in the bone marrow that's starting to proliferate to divide in an uncontrolled manner. And so, you know, gene therapy would be an ideal option for something like that. Okay, last slide here. The challenges, targeted delivery, which I told you about. You can't really control where the gene goes in the body. Until recently, actually. So you might have heard of something called CRISPR technology, which is this really powerful gene editing discovery that scientists have made. Now they have a much better control at which part of the DNA to make the changes at. And that's why I was saying at the intro part here, right? We are at the brink of a very important breakthrough, and if they're able to apply the CRISPR technology, um, uh, across multiple disciplines, then um, they will have overcome this target delivery problem. Okay, but target delivery basically means are you able to deliver the gene exactly where you want it to go? Okay. Immune response, especially using like a vector or something like a virus, some people might have adverse uh, um, reaction to it because after all, it's something foreign, and your body will react against it. So for people who have certain kind of conditions, they are already a little bit immunocompromised. So, introducing virus to them might not always be ideal. Interaction with other genes, that's another issue. When you insert the gene, right? What if the gene goes in the middle of an existing gene? Then that's going to interrupt it, right? Okay. What if in covering up the green light, you accidentally cover up another important gene? What if you cover up like a red light, in addition to covering up a green light, right? So to speak, right? Then that's going to be problematic, right? And cost is very expensive, okay? Right now, these gene therapies are very tailored to the person who have the conditions, and easily we talk about millions of dollars upfront, and then hundreds of thousands of dollars maybe annually to kind of keep up. And so not everybody's going to be able to afford it. Insurance is almost most definitely not going to cover it at this stage. So there is the equity accessibility issue. Um, but it's a very exciting field, and I believe one day gene therapy will be the answer to a lot of the diseases that we have. How to understand mRNA vaccine of COVID? Yeah, so that actually you will have to wait until next semester. Okay, we I have also a little bit of information on that in the immune system lecture. And that is it for lecture number 9. Okay, you survived the hardest 2 lectures. If you understand things in 8 and 9, then congratulations, the hardest part of the course is done. And we must now move on to the last part of the course, which is, of course, lecture 10. Um, Let me just pull that up. I mean, lecture 10 is not too hard. I wouldn't say. Certainly not when you're taking it online, because you can, have some reference to look at. But 10 is quite important because it's like a transition lecture between this course and the next course, right? Next semester, of course, you will all be taking the uh, anatomy and physiology, right? Which is more about the human bodies. And so this lecture, body tissue, is going to connect nicely with that one. So I'm not going to keep you guys too long today. I am just going to go through maybe half of this stuff, and then I'm going to let you go, and we'll wrap it up on Wednesday for lecture 10. So here we go. Stem cells. Right? Stem cells are cells that can cell renew. That means they can make more copies of themselves, self renewal, and they are also able to differentiate into specialized cell type. Okay, differentiate is a big word, but what that really means, it's think of it as like a one-way transformation. Okay, so here you have a stem cell, and under different conditions, they can become different cell types. If we are talking about an embryonic stem cells, like the ones in a fetus, then they literally can become any cells of the body. They can become like our heart cell, they can become a brain cell, they could become a liver cell. Okay, so in this transformation, it's called like differentiation. But it's a one-way transformation. Once it becomes a heart cells, there is no turning back. A heart cell is not going to change back into a stem cells and then go and become a brain cell. You are committed once you are on this particular path, right? So all our cells in the body came from that single cell that was formed after fertilization, right? Sperm plus egg gives you the zygote, and from that zygote, we get all the different cell types, right? So the embryonic stem cells are the cells that is in the embryo, which can become different cell types in the body. Okay, so they are very versatile. After birth, there will still be stem cells. And even in us, like adults, we also have stem cells, but they are less versatile. Okay, we call them adult stem cells, and they are more restricted into a particular category. For example, in the bone marrow, you would have they're called hematopoietic stem cells, okay? Blood stem cells, okay? Blood stem cells. Blood stem cells can become different types of blood cells, but that's it. They can become brain cells. They cannot become, kidney cells. They are restricted within the blood category. And there are different cells in the blood. Okay? There's red cells, right? Different kinds of white cells, and then there are platelets. So all these different cells come from uh, blood stem cells. Okay? You might have some stem cells in your gut, and they would become cells related to the intestinal tract. So these adult stem cells are more restricted in terms of the kind of cells that they can become. All right. Now, I don't know if there are any parents here, but when your baby was born, in Canada, anyways, they have an option for you to store the baby's cord blood. Does anybody know what I'm talking about?
I do.
Yeah, yeah.
So
So,
basically, if they ever get sick and something can't cure it, they can use that to cure.
absolutely, right? So the cord blood, for those of you who don't know, is the blood that is it's a baby's blood basically. That's kind of left over from the umbilical cord, right? Okay, and then when they cut it, they could extract the blood, and that blood contains embryonic stem cell, okay, from the baby. And the idea is like we're not there yet, okay, but maybe sometime in the near future they are able to use the stem cells to grow different organs because remember these stem cells can become any cell type, right? So they're saying, why don't you pay a bit of money and then store the cord blood, and if your kid needed it in the future, they would have the cord blood. They would be able to take the stem cells and potentially treat, certain types of conditions, right? I think it was quite expensive. I mean, I didn't do it, but it's not cheap. All right. Uh, Before this thing was available, have you guys heard of like I think it's called a savior baby. That's like the term. So like sometimes for parents if they have one child and the child has a certain kind of disease and the only way to cure them is to have someone who has a close enough matching um It's called antigens, okay? Matching profile, okay, genetic profile, okay. And if the parents are themselves are not suitable candidates, sometimes they would decide to have like a second child, and then the sibling would be close enough genetically to help out the older child, right? So it's a similar idea, right? It's like these stem cells able to become different cell type and be able to treat certain kind of diseases like leukemia, for example. Okay, so if you're interested, you can look these things out. All right, back to this beautiful diagram, which I actually showed you all the way back at week number one. We talk about atoms coming together to form molecules, right? We went through the whole organelles and we spent, 5, 6 weeks talking about everything that's going on in the cell. Finally, today we are moving up to the tissue level. Okay, and next semester it's all about organs and organ system. Each week we will spend 2 classes looking at a specific organ system, how it works, what happens if they don't work, and learning all the names about that system is really quite exciting. All right, and here are all the system that we're going to be learning, with the exception of the integumentary system. Okay, we don't have time to learn about the skin. It's very important, actually, the skin. It keeps, bad things out of the body, but everything else we will learn next semester. Okay, here are some important cavities in the body. We have the cranial cavity up here in the head. Now, obviously, that's where the brain goes. Okay, and then further down. Okay, we have the thoracic cavity. Don't worry about the things I don't circle, okay? The thoracic cavity. Can I have some organs from the thoracic cavity for here, please? And anything that comes to your mind?
Your heart,
Heart. What else goes in there, guys? The lungs, yes. Anything else? Maybe one or two more things. One, two. Doesn't have to be an organ, could be like a structure.
esophagus. I don't know.
Ah, very good. That's exactly what I was thinking about. Esophagus, trachea, excellent. Okay, so just kind of know what's in the thoracic cavity right here in the chest area, huh? Okay, then there is the diaphragm. Diaphragm. Okay. It's a muscular sheet that helps you breathe, and it separates the thoracic cavity with the abdominal cavity. Okay, so here's the abdominal cavity. Same thing. Can I have a few things here? Just
Big
type them out.
intestine, small intestine.
Sure, yeah, the intestines.
Mhm.
We have the liver. Yeah, a couple people say liver. What else? Maybe one or two more. Bladder. Which bladder? We have two bladders. Gallbladder. Yes, thank you. Okay. Stomach, right? Kidney. Okay. So just know a few of the common ones. I'm not gonna give you like obscure things. Okay. I'm not gonna be like, oh, where is the spleen, right? Like, we will learn about it. But if I'm gonna ask you what's in whatever cavity, it's gonna be obvious things. Pelvic cavity, couple things here, different things for guys and ladies, and then there are some common things as well. But what's in the pelvic cavity?
Oh, your ovaries.
Ovaries, yes,
Mhm.
definitely. Anyone else? Uterus, yes, for a female. Um, and then fallopian tube, okay, okay. Prostate, thank you. I was waiting for that one. And maybe one more that's common for both male and females. Yes, thank you. The urinary bladder. There we go. Okay, so just common stuff. Next semester we will learn all about these things in great details. But right now. That's that. All right, we're gonna do anatomical terms. This one is fun. Take out a piece of paper and we'll do it together. Okay, so this is gonna be uh this is gonna be gingerbread man right here. There we go. Okay. You can probably draw better. Oh boy, I erased the whole thing. There we go. Let's draw it a little bit better. Okay. Very happy. And this is called the anatomical position. Anatomical positions. Okay, so like it's like I don't know if you guys can see me in the camera, but it's basically standing here. You can't see my legs. I promise you, I'm standing, and then your hands are kind of on the side, palm facing forward. That would be the anatomical position. Okay, so whenever we talk about body parts, as in like where they are in relation to one another, we are always thinking about the anatomical position right here. Okay. The arms probably should be a little bit lower here. Let me just try to fix that up a little bit for you. Okay, there we go. Okay, with the hands there. Okay, now you're thinking, like, do I really have to draw this? Yes, you do, because I'm gonna put a lot of stuff on it, okay? A lot of stuff on this thing. All right. Starting with what's on top and what's at the bottom. Right here. Right, this is on top and this is below. If things are closer to the head, we say superior. Superior. All right. What's the opposite of superior? You know it. What do we say?
inferior,
Absolutely. Inferior. Okay. So your head is superior to your chest. Okay. Your nose is superior to your mouth. Make sense? Superior, inferior. Okay. Keep this guy. We're going to come back to it. I have lots more terms to add to it. So here we go. We have some fill in the blanks here. Just take a moment and fill it in. We only have two terms here, superior and inferior. All right. Shouldn't take too long, right? I hope. The hands are blank to the feet. Superior. The knees are blank to the waist. Waist is here, right? This is waist, and then the knees. So knees are blank to the waist. It would be inferior. Elbow, elbow is blank to the wrist. Okay, remember this is not anatomical position, right? Anatomical position has to have your arm down. So elbow is superior to the wrist. Calf muscle, if you think about the leg, okay, calf is like at the back here. That's the calf. And the ankle is here. So the calf is superior to the ankle. Are we together with superior, inferior? Anybody have questions? Or if you have another example you want to throw at me, it's easy when there are only two choices, right? Of course. Let's do more. Okay. All right. This one is we have to draw another person. I'm sorry. This one we have to draw it from the side, okay? I'm not very good at drawing this thing on the side, but let's try. Okay, so here we go. Okay. Let's use a different color. You probably draw the most stuff, right, from my class, if I'm not mistaken. I don't think you guys draw much in other subjects. So having good art skill is good. We're gonna be drawing even more next semester for anatomy. So there, it's in front, and this is behind. In front of is called anterior, and behind is called posterior. Now, there are some other terms that you will see. Uh, Ventral and dorsal. Ventral is same as anterior when we are talking about humans, okay? And dorsal is same as posterior. But actually, anterior means the front, posterior means the back. Ventral means the tummy side, okay? Where your abdomen is, that's ventral. And then dorsal is like where your back is. So that's not always the case, right? Like if you think about like a dog, okay, dog looks more like a mouse, but whatever. Um, this here is anterior. This here is posterior, and then this here is ventral because that's like the tummy, right? The abdomen. And then this here would be dorsal. Okay. So like, it depends on the organism you're looking at. These terms might not always be the same. Like in a shark movie, right? Like you see the shark fin here, right? Anybody know what the name of that fin is? It's one of these terms, okay? Something fin, right? If you have to guess, is it this one, or this one? What's the name of that fin? Shark, guys, like shark. Yes, thank you. That's the dorsal fin. Okay, dorsal fin. It's at the back, right? Okay. So, these anatomical terms are universally used. Okay, back to our very serious discussion right here. This is the second set of questions. Let's take a look. All right. The heel is blank to the toes. Okay, so again, heel is here, toes is here, and you're in anatomical position. So that's gonna be and posterior, right? Posterior. We use anterior or posterior. Okay. Dorsal and ventral, we actually use them when we describe the nervous system, but for now, just stick with anterior and posterior. Okay, biceps and triceps. This here is bicep, okay, and this here is tricep. Bicep, tricep, and in anatomical position like this. Bicep, tricep. What's the relationship? Well, I hope you choose anterior. Hamstring and quadricep. Oh, that one's hard to show. There's a lot of movement today. Okay, so this is the leg, right? Let me move the camera right here. On top, this one is the quadricep. And then right here, that's the hamstring. So, what's the right word that goes into the blank? Posterior, that's correct. All good? All right. What's next? All right, next here. We are gonna go back to this guy right here. Oh, gingerbread man. And we're gonna divide into left and right half. If it's further away from the middle, then we say lateral. And then if it's closer to the middle, then we say medial. Okay? So imagine a line going down the middle. If it's further from the center, from the middle, then it's lateral. It's closer, then it's medial. So the nose is medial to your eyes. Okay. Now, obviously, you can say the nose is inferior to the eyes as well, right? There are multiple correct answers. But sometimes one is more appropriate than the other. It really depends on what you're trying to compare, right? So the nose is medial to the eyes. And what about the ears? Ears are lateral to your eyes. Okay. So what's in the middle? What's the most medial thing? Right? We have the nose, we have the mouth, and then like the belly button, right? That's also medial. Okay, go for it. Midline is the line that goes down the center, right? So just use lateral or medial for this question. So arms are lateral to the midline. Everything is lateral to the midline. Okay, there is nothing more medial than the stuff that are on the midline. Midline, okay? Nothing's more medial than your nose. Nothing's more medial than your belly button. The neck is blank to the arms. Okay, medial. Neck is right down the midline as well, right? Shoulders are lateral to the midline, and the ears are lateral to the nose. Okay? All right, couple more. The next pair of terms are used on the arm or the legs. Okay? It's going to be on a limb. Okay? So on the limb, either the arm or the leg. If it's closer to if it's closer to um the joint, so like at the hip, for this is the hip. If it's closer to the hip, then we say proximal. If it's further down the hip, from the hip, sorry, we say distal. Same thing for the arm. If it's closer to the shoulder, that's where the joint is, then it's proximal. If it's further from the shoulder, then it's distal. Okay. Distal, proximal. Distal, proximal. So it's compared to the joint. All right. Fingers. Fingers compared to the elbow. Remember, this is the joint, right? The shoulder is the joint. The fingers are farther down the joint compared to the elbow. So it's going to be distal. The knees are closer to the hip than the feet. So that's proximal. The wrist versus the finger, wrist is closer to the joint, right? So that's also proximal. Are we okay? I have four more for you and then we're done. Just want to make sure that no one's confused. Remember, there are more than one possibilities. Fingers can be inferior to the elbows as well. Okay. All right. The next two is I cannot draw it on the diagram. I have to go back to the PowerPoint, okay? And it is ipsilateral. That's how you pronounce it. Ipsi and contralateral. Ipsilateral means it's on the same side. Contralateral means it's on the opposite side. So the spleen, which is right here, okay, in case you don't know where the spleen is, the spleen versus the gallbladder. All right, so the midline goes down the middle. So are they on the same side of the body or the opposite side? Well, it's opposite, obviously, from the picture. So we say contralateral. Contralateral. Okay, your appendix is contralateral to this to the spleen. Okay, the stomach is here, and then the liver. Actually, you know what? That's a bad example. People always say, "Oh, I see a little bit of a liver," but I guess like the bulk of it, it's on the other side. So we'll say contralateral as well. Contralateral. Okay, this here is the appendix. Appendix and gallbladder, ipsi, ipsilateral. Okay. Last pair, and we're done. deep versus superficial. Okay, it's exactly what it sounds like. If something is deep, is closer to the core of the body, like on the inside. If something is superficial, then it's closer to the skin, closer to the surface. So bones are generally located blank muscle. Are the bones deeper or are the muscle deeper? Right? That's what you have to think about, right? Like I'm sure you know, bones are deeper, so located deeper then. Huh? Like when you pinch yourself, you're pinching the muscle. The bone is like buried deep inside. Okay, skin are superficial to muscle. Cranium is the skull, right? And is the brain outside of the skull or inside? Obviously, is inside. So we say deeper. Now, you might not know what this is, and it doesn't matter. You just read the description. The pericardium is a membrane that surrounds the heart. We can say the pericardium is superficial because it surrounds it. That means it must be outside. Superficial. Okay. Not superior. Superficial. Okay? Don't confuse the two. Yeah. No worries. That's all. Okay. So practice using these terms because we are going to be using them like a lot next semester. Okay. When I show you a picture of like for example, let me see if I have a quick picture to show you. Six, yeah, over here. Like when I show you a brain picture, like this, then you have to know, oh, this is a like a lateral view, okay, of the brain. Or like if I show you this, this would be like a superior view of the brain. Like, are you looking at it from the top? Are you looking at it from the sides? Like, is it from the outside or from the inside? Right? So you have to know the terms basically. And not to mention you of course have to know them for the final test as well. Okay, so quiz on Wednesday for you guys. Lecture 8 and 9, and then come to class on Wednesday. We will finish up the lecture 10, and then we'll be done the course. Okay, final test next Wednesday. You're gonna have a review on Monday, but we're not gonna have a live class on Monday. Okay, Monday I'm just gonna post a review for you to watch. Of course, if you have questions, you more than welcome to email me. Okay. But this Wednesday will be our last synchronous class. Okay. That's it. Have a good day, guys. See you later.